You're listening to The Chain, a science podcast where we bring what is new in biologics and protein engineering to the community of scientists working in this field. We discuss the latest developments with leaders who are on the front lines of cutting-edge research. On episode 6, Dr. Nicole Liscombe sits down to discuss bispecifics for T-cell engagement with Dr. Paul Parren, who is the Executive Vice President and Head of R&D at Lava Therapeutics. Professor Parren is an expert in antibody research, translational science, and drug development, and will be discussing some of his recent work with T-cell engagers.
Hello everyone, I'm Nicole Liscombe, a Conference Director at Cambridge Health Tech Institute. I'm delighted to be speaking to Dr. Paul Parren. He is Executive Vice President and Head of R&D at Lava Therapeutics. In addition, he is Professor of Molecular Immunology at Leiden University Medical Center. And previously, Paul led preclinical development and research at Genmab. So, Paul, can you tell me something about Lava Therapeutics and the goals and achievements of this relatively new company?
Yeah, sure. So at Lava Therapeutics, we create and develop next-gen T-cell engagers for the treatment of cancer. And we specifically focus on the generation of bispecific antibodies that can engage gamma delta T cells to kill tumor cells.
I know that the concept of T-cell engagers is no longer new. What do you see as the recent advances by academia and the industry in the field?
Well, it's correct that bispecific T-cell engagers have been around for quite some time, and for a long time, progress has been really slow. However, advances in the bispecific antibody production that have come about in the past number of years, mostly at companies, but also that combined then with clinical successes in immuno-oncology, really have sparked enormous interest and investment in this field in the past few years. So this is really reflected also in the 40-odd T-cell redirecting bispecific antibodies that are currently in clinical development in hematology and solid tumor indications, really showing how fast this field is moving. And many others are in preclinical stages of development. Well, for those detailed information can be found in my review in bispecific antibodies in the August issue of Nature Reviews Drug Discovery that just came out.
Wow. Oh, thank you for that. Excellent. That's really exciting. So what do you see as the big challenges still being faced?
Now, most of the bispecific antibodies in development that activate T cells via CD3. And despite their promise, and as I just mentioned, the many molecules in development, targeting T cells via CD3 does have a number of disadvantages. One includes high toxicity, and is particularly seen for targets with broad expression. And that really then limits the addressable anti-target space. So we need to constantly be looking for good targets for this to combine with this technology. And a further challenge is insufficient activity. So particularly in solid tumors, the responses have been promising for some targets, but less so for others. And at least in part, this can be explained by the fact that CD3-based T-cell engagers really activate all T cells, so including T regulatory cells that actually inhibit the anti-tumor activity. So we further need to improve safety and increase activity of these compounds or of this particular class of molecule.
We hope you're enjoying this episode of The Chain and wanted to take a minute to share some exciting news in the world of protein engineering. The 11th annual PEGS Europe Conference is taking place November 18th through the 22nd in Lisbon, Portugal. You can learn more about the meeting at www.pegsummiteurope.com. That's www.pegsummiteurope.com. Early registration rates apply until September 6th, so head over to register with the key code POD100 to save $100 on registration. That's POD100. We hope to see you there.
Interesting. So what are you doing currently at Lava Therapeutics to overcome these challenges? And what are the advantages of your approach over existing ones?
Yeah, so one exciting approach is to move to generate bispecific antibodies that are designed to only activate specific effector cell subsets. So to make much more specific compounds. So at Lava, we generate bispecific antibodies that engage gamma delta T cells, and more specifically the Vγ9Vδ2-T cell subset. Now, this is a relatively homogeneous population of pro-inflammatory effector T cells, and these cells also already have a natural ability to recognize and kill cancer cells. So the presence of these cells, for example, in a tumor microenvironment, correlates more strongly with patient survival in a wide range of tumors than any other lymphocyte subsets. So we really believe that specific targeting and activation of these gamma delta T cells with our bispecific T cell engagers will lead to the development of more efficacious and also more safe treatments for hematological and solid tumor indications. Now, Lava is a startup company, and so we're gearing up to have our first product in the clinic by the end of next year.
Well, that's really, really impressive. And we look forward to hearing more about it at PEGS Europe. So tell me, Paul, how many times have you been to PEGS Europe and what are you looking forward to this time?
Yeah, I've been thinking about that. I've definitely have visited PEGS Europe several times over the past number of years. I must say I always enjoy the excellent presentations and also the outstanding possibilities for networking with experts in the therapeutic antibodies and related fields.
Wow, it's really, really good to hear that. So thank you very much indeed.
Thank you for joining us on The Chain. Tune in next time for more conversations about science, research, and exploring the world of protein engineering.